1,000-year-old bones discovered in caves in Eastern Europe. They are reconstructing the genome of direct ancestors in our lineage, in addition to independent hominin branches, including Neanderthals. Remarkably, the exact same two FOXP2 proteins that differentiate modern humans and chimps are shared with our direct ancestors, and surprisingly, also our Neanderthal cousins. There's debate about whether Neanderthals possessed anything like human language, but this shared genetic link will be informative once researchers nail down how long ago humans and Neanderthals diverged in our evolutionary past. The estimates are currently anywhere between 300,000 and 600,000 years ago, and once scientists pin down the dates more definitively, they'll have a much better estimate of the minimum number of years that we have carried the human form of the FOXP2 gene. This is because humans and Neanderthals are not totally identical with respect to the FOXP2 gene. There are some subtle differences in binding sites where the gene connects to other molecules, and these differences may be key in understanding what makes human language unique. In addition to looking to the past to better understand how FOXP2 relates to language, researchers are also focusing on the present. The Human Genome Project that began in 1990 was finally completed in 2003, and it provided the world with the first ever full map of the human genome. This incredible feat has opened the door to scientists and entrepreneurs alike to figure out faster and cheaper ways to map the genome. We're now in an age where for about the price of a transatlantic flight, you can purchase a full genetic sequence of yourself. And for a much smaller investment, you can pay companies like 23andMe or Ancestry.com to provide a report of your genes as they relate to common genetic variations. With this sort of technology taking off, researchers believe that in a few years, we will have a new wealth of genetic data to map genes onto normal variations of human language. Researchers like Simon Fisher, now at the Max Planck Institute for Psycholinguistics in the Netherlands, they're hopeful that this new information will reveal more subtle mappings between FOXP2 and language. For example, perhaps there are mutations of the gene that correlate with enhanced vocal phonological abilities, as with auctioneers, singers, and polyglots, like speakers of multiple languages. This is more than just a mere novelty. This sort of work could be invaluable for the future medical treatment of individuals with speech production problems. Beyond FOXP2, there's hope that we'll discover new gene variants that connect to different aspects of language. Remember, language is a complex system. The FOXP2 gene links mostly to phonological and some syntactic aspects of language, but other aspects of language are unrelated. Might we also find genes that map onto these other parts of the system? There are cases of other genetic disorders where some aspects of language are disrupted, but others are spared. For example, Williams syndrome is a disorder caused by the deletion of 26 to 28 genes on the 7th chromosome. And this causes a unique psychological and social profile. Carolyn Mervis of the University of Louisville did some of the pioneering work on Williams syndrome, and she describes a disorder in which there are several distinct cognitive deficits in math and spatial abilities, but a relative sparing of some language abilities. In particular, phonological and semantic abilities are close to average in these individuals. Contrasted against their other impairments, these skills really stand out. In fact, if you met a child with Williams syndrome, you might be superficially struck by how articulate and well-spoken they are. But this belies some deeper language problems. Their syntactic abilities are lower than their phonological and semantic skills, and their pragmatic abilities are extremely compromised. This deficit in pragmatics comes as a surprise to anyone who's even briefly interacted with a person with Williams syndrome. On first impression, they come across as quite gregarious and interested in others, but a closer look reveals some significant social deficits. For one, they have great difficulty following the thread of conversations, In fact, they're known for making many non sequiturs which are novel and interesting, but demonstrate poor understanding of the usual social give and take of a conversation. And 2, formal tests of theory of mind, which assess perspective-taking ability, reveal that their ability to shift to the perspective of others is also quite poor. This means that they have a very hard time knowing what other people know and don't know, which is a key pragmatic skill. These pragmatic deficits are reminiscent of problems also seen in individuals with autism spectrum disorder, or ASD. The National Institutes of Health describe children with ASD as often socially withdrawn and having limited ability to successfully communicate and interact with others. Often these individuals have difficulty developing language skills and understanding what others say to them. Many also have difficulty communicating nonverbally, such as through hand gestures, eye contact, and facial expressions. In contrast, some people with ASD have incredibly rich vocabularies and very sophisticated syntactic skills. This again suggests an interesting disconnection between semantic and syntactic abilities from pragmatic skills. Here's a nice example of the dissociation in the words of Temple Grandin, a scientist and author who has written extensively about her own experience with ASD. Social relationships have been learned solely by intellect and use of my visualization skills. All my thoughts are in pictures, like videotapes in my imagination. When I encounter a new social situation, I can scan my data banks for a similar situation that I can use as a model to guide me in the new situation. My data banks and social skills are also filled with news articles about diplomatic relationships between different countries and an archive of previous experiences. I use these scenarios to guide me in different situations. From this excerpt, you can see why Grandin has described herself as an anthropologist on Mars. She's taking a very intellectual and objective approach to understanding social complexities and human interactions and language that comes so naturally for so many others. Currently, researchers are searching for genetic clues or causes of autism. We know from twin studies that ASD is highly heritable, but it's been hard to pin down the exact genetic mechanisms. Part of the problem stems from the fact that unlike verbal dyspraxia and Williams syndrome, ASD is a much fuzzier disorder. Because it spans a continuum, it's not all or nothing, it's hard to connect to a clear genetic cause. Once we better understand this genetic link, we'll start to piece together the different genetic mechanisms that underlie all aspects of the language system, from phonetics all the way up to pragmatics. So, to sum this section up, there are 2 main take-home messages. 1, there are no single genes to explain all of language. Because language is a complex system, we must recognize that different aspects of language are affected by different genes and different gene networks. And 2, it's likely that much of the machinery of language is built on genes that are shared with other species. When we talk about evolutionary conservation of genetic material, we're talking about homologies. A homology is a structure or a process that is shared among species due to a common evolutionary ancestor. They can be at the gene level, as with FOXP2, and also at higher levels of organization, such as cell types or organs. As we move higher up from the genetic level, it's clear that humans and many other animals share homologies related to language. For example, the chimp brain and the human brain are anatomically very similar. We both share the same basic hemisphere and lobe structure that I introduced in Lecture 3. Even Broca's and Wernicke's areas have homologous structures in the left hemisphere of chimps. And just as with humans, these regions are involved in the production and comprehension of vocalizations. Interestingly, we also share a network of neurons in these regions that are part of what has been called the mirror neuron system. Mirror neurons were first identified in the monkey prefrontal cortex by Giacomo Risolati back in the mid-1990s at the University of Parma. These neurons have the interesting mirroring property of firing both when a monkey reaches for an object and also when the same monkey observes another monkey or human reach for the same object. One interpretation of these neurons is that they are basic cellular mechanisms for understanding the intentions of others. Interestingly, two of the main neural hubs of the system in humans are the inferior frontal gyrus, where Broca's area is located, and the medial temporal gyrus, just below Wernicke's area. This means parts of the brain that are mechanisms for language are also involved in understanding the intentions of grasping actions. This link between hand movements and language has led some researchers, like Michael Corballis of the University of Auckland, to theorize that spoken language systems emerged from gestured communication systems in our evolutionary past. We'll talk more about gesture in a couple of lectures, but I bring it up now just to show you how language builds on structures that are shared across species. Now, even though there are many homologies between humans and our closest primate cousins, there are also some major differences. For example, the human brain is much bigger than a chimpanzee brain, even when adjusted for relative body weight. Recent research by a leader of the Human Genome Project, David Hostler, suggests that these differences may be driven by a unique family of genes called Notch2NL. These genes emerged after humans split from the ape lineage several million years ago. This genetic difference may also be responsible for the number and complexity of neural connections in the human brain. One recent estimate is 100 trillion connections, trillion. Just to get some perspective, that number is hundreds of times greater than the number of stars in our galaxy. More than sheer number, the way that neurons are connected is crucial. One of the novel aspects of the human brain is how many connections it has between new and old brain structures. In all mammals, the newest part of the brain, called the neocortex, lies on the surface of the brain, and older parts, like the subcortex, lie deeper beneath the surface. Both structures are very old. It's estimated that the neocortex was added on top of the subcortex when our distant mammal ancestors diverged from reptiles during the time of the dinosaurs. The neocortex is where most high-level cognition occurs. Indeed,